Коллеги, доброе утро. Очень рада, что сегодня много людей проснулось вовремя, пришли к нам вовремя. Меня зовут Ильназ Климовская, я работаю в компании Novartis Pharma, я менеджер по продукции, занимаюсь препаратами против глаукомы. Вот. И сегодня я с большим удовольствием хотела бы вам представить человека, который вам расскажет про подход компании Novartis к персонализированной медицине. Это Кришнан Раманатан. Кришнан – это директор по науке в компании Novartis Pharma. У Криша на самом деле огромный научный опыт и, может быть, даже про свою карьеру и, так скажем, бэкграунд Криш сам вам расскажет. Поэтому давайте мы все поприветствуем Криша и ждем интересной лекции.
So I'll speak in English. I will not embarrass myself and you with my Russian. So thank you for the opportunity. My name is Krishnan Ramanathan, and I had the pleasure of working with Ilnaz for a few years until she found some more interesting opportunities. So I will speak about myself a little bit before I get into this topic, because this is a collection of many topics as such, because it's not easy to speak in a short time about drug discovery and the future of medicines, because it's a very deep topic. So a little bit of a story about myself. I've been in Russia for about 2.5 years now, and I head the research and clinical group in Russia here for Novartis. Well, we say research and clinical, but most of the development work here is clinical development, is clinical trials. And we have today over 40 ongoing clinical trials in Russia for new medicines that we want to bring in here, because it is a part of the regulation that we have to test all new medicines in Russian patients. So that's part of my job. Another part of my job is medical affairs, which is making sure that the right medical information is available for new medicines that we bring in here. And so that's mostly what I do today. But if I turn the clock back close to 20 years, I was a student, I was a PhD student working on hearing sciences. So it was neurological work. It was in the Department of Biomedical Engineering and Neuroscience in Johns Hopkins University. And I was just telling Alexander about my work on ion channels. So the work I was doing was to actually understand some of the fundamentals of hearing science. So when you see sound or hear sound in your ear, what happens is the mechanical energy is transduced to electrical energy, and some part of this process utilizes ion channels, almost like bandpass filters. So these ion channels are sensitive to certain frequency of stimulation. And some of the previous work in my laboratory there and in my advisor had described how these ion channels are able to get this electrical properties by using different types of splicing. So I used a kind of a model to describe that, and we published it. Interesting thing is, after I finished my publication and left academia, I have not worked on that any longer. So now that's 18 years old, and somebody else is referencing those information and developing the science. And in some ways, that is the beauty of science, that it continues through others. When I did my work, I could speak to maybe 6 people in the whole world who understood what I did, who actually had interest in what I did. But now there are more people, because some of these basic properties of structure-function relationships in ion channels is much better known today than it was in the past. So since then, I've been working in industry. I've worked in 2 big pharmaceutical companies. I started my career in GlaxoSmithKline, also in R&D in the United States. And then I spent a couple of years working in a small biotech. And this was a company with 1 molecule trying to make that into a medicine. And that was a very interesting experience. And I say interesting experience because the medicine didn't make it. So all I have today is the experience and not the medicine. So this is also some of the challenges of working in drug discovery, that not all molecules that you discover really become medicines. They are interesting molecules scientifically. You can characterize them, you can understand them. But in order to convert them to medicine, there's a lot more things that you need to do. So, and after that, I've been now working in Novartis for 10 years in research and development in different functions. In some ways, it is very different from academic world, but at the same time, our mission is the same. The academic world is looking at more of characterization of physiology, receptors, drug targets, and in the in the pharmaceutical world, we try to take these and make a business out of it. So take a target or take a molecule, make it into a medicine, and find ways to commercialize that and use the profits to further invest into R&D. So with that kind of background, I will give you, this talk is in actually 3 parts. So the first part is just to give you some background of how medicines are developed today, because it's quite complex, but at the same time very interesting. So I give you some background of how we develop medicines. Then the second part, we talk about how we can personalize medicine, and this is not a new topic. And I'll give you what my take on that is. And the third one is, can we actually cure diseases? And we are actually at a point where we can attempt to cure many diseases. So I'll try to talk about that. Is my pace okay? Should I slow down? Or is this okay? Okay, thank you. So drug discovery, this is a slide that I show to many people. We start with a target, and today some of these targets are well validated because we've sequenced the human genome. We know what proteins they are, what receptors they are. And once we identify a target, which is a druggable target, where we know we can influence that target and change the course of a disease, we try to find a drug, and then it goes through many periods of testing. Now, the beauty here is that you see we start with about 10,000 compounds, and most of this is true with chemical entities. Some of the numbers are different for biologics because proteins are characterized a bit differently. So we start with 10,000 compounds. So we have a library. It's almost like a book library with boxes of different chemicals. We take some of these chemicals, we test them in high throughput screening, and some of them make it into in vivo testing and some into the clinic. Often what happens is the molecule that is in the library describes a series in chemistry, and they need to be modified a bit to fit into the receptor, to make it more easily manufacturable for various purposes. And when we bring it to human clinical trials, this is where the majority of the investment is. Here's where we are spending a lot of money, because this is only about 40 years old, the way we conduct clinical trials. Of course, drug discovery, we were inventing medicines since man discovered aspirin or even before that. But clinical trials are very recent, and the investment it takes to do a clinical trial is huge, is enormous, because it involves patient ethics and consent from patients, and we have to do it in multiple countries. So here's where most of the money is spent, and then eventually we get a medicine. So it takes 10 to 15 years, and Russia participates here in many of these that we accomplish clinical trials. We work in about 400 sites in Russia with Novartis for doing clinical trials. Now the challenge here is, you see the investment, it keeps going down or up, and we spend about 2.6 billion. Now this number is highly disputed, it varies, it's not a single number. The way it is calculated is, you look at public records of what companies are spending in R&D. And Novartis spends about $9 billion in R&D every year. And with $9 billion, we can get 3 to 4 new medicines, not more than that. And you look at how many medicines are being approved, and you do this average over 10 to 15 years, and you calculate this number. So it's a very approximate number, but at the same time, a powerful number, which describes the complexity of drug discovery. And you see, now the complexity is further enhanced by the cost of developing that medicine has been increasing over the last many years. When the FDA was first established in 1950s, 60s, post the World War time, all you required to show was that a medicine caused no harm to humans, or a chemical caused no harm, and then it was approved as a medicine. So if salicylic acid caused no harm, and you found a use for it, it was approved for that use. But now it is not sufficient to show that an antibody just causes no harm and it's good for man. You have to show that it actually shows a benefit in a certain disease, and that process is getting more and more complex as we discover more and more new medicines. But what is the benefit of all of this to society is we're all living longer, and hopefully we will continue to live longer, and many of us will live over 100 years old in the future. Now, we have in Novartis and many companies, we have a very scientific approach to do this, because we know the human genome, we know what are the targets, we know what potential indications you can have. Once you identify a molecule, so this is one of the molecules that I worked on, so I like to show it. So you know the pathway of immunology, of how Th1 receptors or Th17 receptors produce, or 17 molecules produce interleukin-17, and interleukin-17 is implicated in many diseases or many indications. So we can then start in parallel a lot of work doing research in psoriasis, rheumatoid arthritis, ankylosing spondylitis, in asthma, in diabetes, because we know that this pathway or this molecule has properties that could potentially help many of these diseases. So all of this work is done in parallel, and you know why we do it in parallel is because the expense is too high. If we stop to do it in series, and we lose out our time from the patent. So the minute we submit a new molecule as a patent, we have about 20 years, and after that anybody can make this medicine. And in fact, the drug prices do not reflect the cost of making it. The prices reflect the cost of researching it, and this cost is extremely high. So if I look at it, 70% of the cost of any molecule in developing it is in clinical trials. And this cost is continuing to increase over the future, and that's why medicines are expensive. And this shows that timeline of why we work in parallel. So we spend many years actually investing in research and clinical trials, and then these sort of reflect our sales, if you will. What a pharmaceutical company recovers from the investment, but then the benefit to society is much longer. So I can think of Novartis invented Gleevec, which is a molecule to treat cancer, certain types of blood cancers. Now, we had a 20-year period within which we commercialized Gleevec, and many of these patients with chronic myelogenous leukemia, at that time when Gleevec was invented in 2000, they had a 5-year life expectancy, or 6, or maybe 7. Today, those patients are still alive. And because Gleevec has given them a new promise, it made a fatal disease into a chronic disease that you can live with CML and continue to treat and keep that under check. So Gleevec, many patients are still using it, but now it's generic. So anybody can make Gleevec. And in Russia, there are many copies of Gleevec available for a much lower price than what we sold it for. So the benefit is huge, but the inventor, many of you may be inventors or trying to invent new medicines or new science, the benefit to the inventor is just small. So you have a small, limited opportunity to capture, and you need to work on it quickly. Now, what is happening to this investment is that we have to continue to make more and more complex clinical studies. And this is an example that shows some of you who are familiar with how we write protocols for clinical study. Some of these protocols can be 60, 70 pages. So we give these to doctors who treat patients in the clinic, and they have to follow this protocol to identify what are the biomarkers or what are the tests they need to do for these patients. And these clinical studies are getting very complex. Each patient, when they visit a doctor, have to spend 4 hours, 6 hours sometimes to provide all the data, some blood work, some MRI, other tests, in order to have one visit for a clinical trial. And what is the implication for us is the cost becomes more and more, and the complexity of these trials. A single clinical study report today from, say, we do a study with, say, 1,000 patients. I've seen a clinical study report that has 18,000 pages if you print it. And many of this is just appendix, because each patient produces hundreds of pages of data. They have their first visit, you take their body temperature, their weight, everything, because you have so much data that you can analyze. You may change their blood pressure, but if you increase their body weight by even a few kilograms, that's a problem. That medicine will not get approved. So today we have to analyze everything altogether, including we collect their DNA. We don't sequence all the genomes, but we keep them, because in future maybe we could sequence it cheaper and understand specific aspects of personalization, which I will touch on later. So this is the complexity that we are going through. And what's happening is that complexity is squeezing the industry, which is us, for the opportunity to commercialize. And some of this you see manifested in disputes of the patent. We try to extend this life to say, can we get an additional few years of patent protection? And some of the regulators are helping us to provide incentives for the industry to develop new medicines, because otherwise, if you have a very rare orphan disease for which it takes 15 years to develop a medicine, we will not be able to recover any money from it. So we would say we won't develop the medicine as an industry. So the regulators say that you can get an extra 7 years of patent life, or an extra 6 years, depending on which country, of protection of your data, so it cannot be copied. And that provides an incentive to develop new medicines. But this is the challenge we face, to develop medicines quickly. And if you can come up with ways to develop medicines more quickly, we are extremely keen on listening how we can do this. So that gives you a kind of backdrop of what I do, what Novartis does as an industry in developing new medicines. I say that altogether in Novartis we have close to 20,000 people in research and development, between research and development. In Russia, we have about more than 100 people doing clinical trials in Novartis who are helping execute these protocols by collaborating with institutions and doctors. So now let's talk about personalization. Now, I was asked to talk about this. I'm not the expert on personalized medicine, but I have some ideas. So I will try to give you some background on this, and then you can challenge me later on if there are any interesting questions. Now, I did a quick study on what is personalization of medicine, and I have worked on some aspects of it, so some of this is my own research. This is not a new topic. In the very old ages, there have been many rituals where, if I look at the historical medicine, we never treated a disease. We only treated the patient. So when you treat a patient, it is personalized medicine, right? And I can reflect on the Indian Ayurvedic medicine. I've been to an Ayurvedic doctor in India, and they don't ask you what disease you have or what complaints you have. You go and you tell them a story. They test your body, your pulse, your weight, your eyelids, whatever they want to test. And then sometimes they can even diagnose what pain you have before you tell them. So they pay so much attention to each patient that it is personalized therapy. What they give you eventually is a mixture of some leaves and herbs. It's like black potion that you drink or oil that you apply if it is pain. And it was completely personalized. It doesn't have a logic or a scientific approach like we do today for testing in trials, but it has the experience element where they could tell what this patient wanted. So personalized medicine is not new. So the Indians did it, the Chinese did it. Then, in Greece, there was practices. Now, the first observations, interestingly, I noticed that Pythagoras observed that some individuals developed fatal reaction after ingesting beans. And this was the first evidence of pharmacogenomics, if you will, or pharmacogenetics, that some individuals had a big reaction, and it may have been due to either gut bacteria or their own digestion enzymes, and some didn't. And this was an observation that was historically recorded. Today we know that this is actually pharmacogenetics. There are some people who react differently to different medicines than others. But somewhere after this, the start of modern medicine in some ways destroyed the personalization because it made it into a more scientific concept where we said we will find a disease and we will treat it for all patients. So we will take pain and we will give you a pain medication for anybody that has pain. It doesn't matter what kind of pain you have. If you have fever, we will find an antipyretic agent. And most of these modern medicines that use today's technologies or surgical, more precise approaches have taken off this personalized concept and have worked on disease categorization. In some ways, I think we are going back to a personalization because we know so much more about this diseases than we knew in the past. And we know that some of these diseases, although we call them a single type of disease, have many subtypes, have many etiological bases for these diseases. So I give you one example. So I look at, say, a very complex disease like obesity. And it is complex because in some ways it has to be extremely personalized. But as an industry today, we are trying to come up with a single medicine that can treat obesity. And that's a challenge because you can never find a medicine for 30% of the population who could be obese in the future. So different aspects of personalization. One aspect would be you select the population, you find out, you choose who may respond to certain medicines. And if some of you have followed the obesity angle, there are a few medicines. There is one that suppresses your appetite. So those people that have an appetite suppression problem should take that. There are some that have digestion issues or that have fat absorption. And there are today, I don't know what the name is, it's Orlistat or Zenical is available in Russia, I think, marketed by Roche. And that actually reduces the absorption of fat. Now, it has many side effects because if you don't absorb fat, it comes out somewhere and it's not interesting when it comes out. So you can choose the patient population, you can find out, and that's a level of personalization. Now, this is very gross. It's very broad scale. It's not personalization to the individual. Another approach is dose selection. And this, in obesity, is common, but it's more common in other conditions where the dose is extremely critical. For example, those that have blood disorders of clotting have to take a certain medicine which is called warfarin. And warfarin interacts with many things in the body, many enzymes, which we have different proportions of, and you have to titrate the dose to each patient. So you go through a clinic where they give you a little dose, then they check your clotting ability of the blood, and then they increase it or reduce it. And this happens to many medicines today, especially when we are trying to give medicines that have a toxic limit, either with bleeding or other toxicities. Another is patient stratification, and I talked about this with you. You can choose different types of approaches for personalizing the medicine, especially if it is obesity. You can have things that help with gastric emptying or with satiety, and you can actually find some of these out by asking a lot of questions. But that is the challenge today. You need to spend time with the patients and ask these questions. Now, this is the most interesting thing in terms of pharmacogenomics, and this is when we look at a population response, and we call it POP-PK, population pharmacokinetics. If I give a same dose of a drug to 100 patients, and then I look at how many have 50% blockade of the receptor, I will have maybe, say, 20% of the population has this kind of blockade. Now, that may hide a lot of data underneath. If I go into the individuals, I will see that some of them actually had a higher blockade, and some of them had a lower blockade, or they were higher responders or non-responders. Now, what I see is a population response would be something in the middle. And there have been many medicines that have been approved based on a population response without looking at the data underneath. And you will never know it whether you are a non-responder or you are a responder unless you test yourself very thoroughly. And there have been medicines. There is one example of a platelet aggregation inhibitor. I'm trying to think. The trade name is Plavix, clopidogrel. And clopidogrel has arguably 30% of the population do not respond to clopidogrel. Now, clopidogrel in a very, very large clinical study was shown to be 8% better than aspirin. Yes, so you can take aspirin for preventing heart attacks, or you can take clopidogrel, and it's 8% better. Now, do you personally benefit from clopidogrel? You don't know. You could be a non-responder. So it requires a lot of analysis. Now, the challenge is the cost of doing this analysis is quite high. To do the blood chemistry on each individual, to look at their platelet aggregation and will they have a heart attack, this is a challenge. So personalizing an already approved medicine is a challenge. The same thing is with the diet interactions, and I spoke about warfarin before. And warfarin has a specific interaction with vitamin K. And vitamin K is available in many forms of diet, especially in green leafy vegetables and all of these. So when you have a high load of spinach and you take warfarin, it somewhat inhibits the effect of warfarin. So you need to somewhat tailor the diet of the patient, ask them not to eat it, or tailor the dose of the drug based on what diet they eat. And this poses another challenge of personalization because we don't all eat the same food. Now, just for obesity, many people have come up with a complex algorithm. And this is not a personalization, but this is an approach. You can do a microbiome test. You can do a blood test. You can ask a lot of things in questionnaires, anthropometrics, waist size, height, body mass index, all of these, food diary, what you take. You put all of this into a nutrition predictor. You look at glycemic responses, what their enzymes are, how their pancreas are functioning, and then you come up with some personalized approach to this. Now, this so far is a more empirical approach to personalization, where you have medicines that are approved, the diet patterns, some of these anthropometrics, and then you can empirically personalize what each person is getting. A more complex approach to personalization is what this would be. Because today we have the ability to do all these tests. You can actually do single nucleotide polymorphism genotyping. If you knew what receptor constituted something for the patient to become obese. You could do diagnostics plus therapeutics. You could stratify many of these in clinical trials. Now, all of these things can be used. And in the end, you're trying to personalize for each person what therapy they get. Now, this is a challenge. When we've attempted many times to do personalized medicines in Novartis. And in the discovery and development phase, it is a challenge because you cannot do a clinical trial with one person. You cannot, because you need to control the study with somebody else. So you need a twin to see if they have a different response. So you have to do a population study to show that a medicine has a benefit over some sort of standard of care. But you can collect a lot of data that can help you personalize this. And this is what we have resorted to. In one of the examples that we have here is we have come up with a point-of-care diagnostics. And this is an example for a medicine in asthma. So asthma can have many subtypes, but essentially what happens in asthma is you have airway constriction of your lungs, and you're not able to breathe with the normal lung volume. And the airway constriction can come from a result of antigens that are coming from the atmosphere for which you have an inflammatory response. Now, one part of this inflammatory response is immunoglobulin E, IgE. And the level of IgE in your blood can show how much of an inflammation you have from topical inhalation of air. So we have, today what we do for this is that we draw blood from a patient. We send it to a lab for testing to detect level of immunoglobulin E. And then once we understand there is a high level of immunoglobulin E with symptoms, and then we look at the body weight of the person, how much distribution of blood they have, we can prescribe a dose of antibody that blocks immunoglobulin E. And thereby it can reduce the inflammation, reduce exacerbation of asthma, and help the patients. So that's a medicine that we have, which is immunoglobulin E antibody. And in order to prescribe this antibody, we need to know what is the level of IgE in the blood. Now, this process is quite complex because you have to draw blood, send it away. We have come up with a point-of-care diagnostics that can help personalize the immunoglobulin test. So I can take your blood and put a droplet, actually 30 microliters, on this card and scan it. And this uses a piezoelectric technology to detect the level of immunoglobulin E. That is the only thing it can do, this test. But that is the only information we need from this. So what we have today is, we call this equipment NIJI. It's still not available in Russia. It's available in Europe. We're trying to bring it in here. So you can measure the total level of IgE with a very small blood volume, 30 microliters, and in about 12 minutes. Now, some of these are very interesting for you to look at as interesting students looking at biotechnology space. This personalization with diagnostics is an area where to approve these equipment is much more easy than to approve the medicine that we have, omalizumab, which took us 10 years to develop and approve. But this diagnostics came from a small company that we acquired in UK. It's in Kent. And there are only about 90 people working in this company, and they built the equipment. And we acquired this test because it helps us with our medicine. And as you can see, you can do it very quickly. Now, what happens to asthma patients? Now, if you can do this diagnosis and you can test the level of IgE, what if they don't have enough IgE? They still have asthma. So now then this drug is not going to work with them. So we can tell that we don't give this drug. We don't waste the patient's money or the healthcare's money, or for any reason, toxicities with the drug can be avoided. So as a company, as a healthcare institution, we're looking at asthma as different diseases. So the whole, the respiratory society of doctors, of pulmonologists, researchers are trying to reclassify asthma into different diseases so that we can give them the right medicine. Because previously, if you had asthma, all the doctors would do would to symptomatically try to increase the airway space so that you can breathe better, which was fine. It's good for relief of symptoms from asthma. But to change the underlying etiology to either block immunoglobulin E or to look at eosinophilic asthma, we have another medicine today where we can block the release of cytokines from eosinophils. And that all of these lead to airway constriction and mucus that manifest as asthma. So I think as we go in the future, asthma may be substratified or classified into small types of asthma, which can be, eosinophilic or immunoglobulin-driven or innate. And we can try to block the trigger of what is causing the asthma and not simply try to, increase the airway size, which doesn't care about anything that happens in the cytokine and is simply looking at, providing relief of symptoms. So this is a challenge. If we look at what we've done in the past, we've taken the patient, instead of treating the patient, we now treat the disease. The disease was too broad, and now we are sub-segmenting the disease to many minute segments. Now, asthma is not as challenging as another disease, rheumatoid arthritis. And I've worked on some of these medicines in the past as well, and our understanding is that not all arthritis patients have the same type of arthritis. And rheumatologists in the past, 20, 30 years ago, used a small battery of medicines. They had, for example, methotrexate, which was used to treat all forms of arthritis. They had painkillers that they would give you first, and then they would track your progression to see if you needed more medicine. And methotrexate essentially was able to help many patients. But as we understand the fundamentals of arthritis, rheumatoid arthritis is different from a patient who has psoriasis or psoriatic arthritis, is different from spondyloarthritis, is different from gouty arthritis. And all of these are different subtypes. And I don't think I'm even expressing future types of arthritis that we will only know when we study the etiology of what is happening. Now, all of these boxes are actually medicines that are in discovery or approved for treating arthritis. And all of these have been approved within the last, I can say, 20 years, because most of them are biologics, and we've come up with so many medicines. Now, you can go to see a rheumatologist if you have pain, and the typical symptom for arthritis, you have 6 months of pain that does not go away with painkillers, normal painkillers that you can take. Then you should see a rheumatologist. Then the rheumatologist will now look at this map of what can they give you, and they have a choice of so many medicines, and they have to do a lot of tests to find out what type of arthritis you have and what medicines work for you. Now, my personal experience working on secukinumab, which is an interleukin-17 antibody, was that initially, from all of the research we thought we'd understood, we thought the most benefit will be in rheumatoid arthritis patients. And so we performed very large studies in rheumatoid arthritis patients to show that the medicine was good, was effective, was no better than any of these medicines. So from a benefit to society perspective, why would you stop taking any of these and switch to secukinumab? I could take adalimumab, etanercept, golimumab, infliximab. So these medicines were as good, and they've been approved for more than 10 years now. So we stopped the research. We said we need something where secukinumab shows better efficacy than existing medicines. So we need to understand the pathway and where interleukin-17s are expressed at a high level. Now, interleukin-17 is very difficult to measure because they go through transient inflammatory phases. So unless you find somebody who's under acute inflammation, it is like C-reactive protein. You don't get high levels of interleukin-17. And it can be secreted locally. Psoriasis patients can have interleukin-17 under the skin, arthritis patients near the joints, spondylosis patients near spinal joints. So to measure local concentrations of this was quite hard, challenging. So the only way we could do this was by doing clinical studies, by giving it to these patients to see who responded better and trying to understand the subtypes. So now we have different approvals. We have secukinumab approved for types of arthritis like psoriatic arthritis, but it is not approved, or rather we did not submit it for rheumatoid arthritis because the benefit is not big. But for the other forms of arthritis, it is actually better than these medicines, and that's why we decided to submit the approval. So this is a level of personalization where we are taking a disease, arthritis, a broad definition, and segmenting it into many small subtypes of etiologies that constitute it, and poses a good challenge, but also poses a good experience for all of us to develop new medicines and benefit patients in society with this. Now the final part of my talk, can we attempt cures? And over the last, 18 years that I've worked on in this industry, I've learned a lot. I didn't come to the industry with any knowledge of treatments or cures, But now, I think, where I stand now, I think that cures are possible. Cures are possible in many diseases, and in fact, the only things that will stop us is ethics, science, and investment, our own creativity, and who's going to pay for these things. How will society pay for these things? So let's see what are the possible opportunities. One of the types of cures that we've been attempting is utilizing the immune system of the body, because the immune system of the body is extremely interesting. It's evolved over so many years, and it's perfected this art of recognizing foreign things and attacking them. And all we have to do is now train the immune system to attack certain diseases and target them. Now, how do we do it? How do we train the immune system to do a task that we want to do? So an example is this technology called CAR T cell, or chimeric antigen receptor T cells. So here's an example of a tumor cell, and this is an immune cell or a T cell that we can use to target the tumor cell and to kill them, or to apoptose them in a more scientific word. But basically, you can stop these tumor cells from dividing by using your own immune system. Now, how does this work? So, for example, you could have a type of B cell lymphoma. And what is that? Your immune system has different types of cells, and the B cells are a type of cells. And in a state of lymphoma, these cells are dividing too quickly. And so you need to have a certain level of proportion of B cells, T cells, platelets, red cells, all of these. Now, if your B cells are 1,000 times higher than normal, then that's cancer. That's lymphoma. Now, with a lot of analysis, we can find out that these B cells that are cancerous have certain receptors that they are expressing. And some of these receptors we've understood very well, and there are drugs for these. CD20 is one. CD19 is one. Now, CD20, we have medicines for that, as antibodies to conjugate with them and to deplete these B cells, and they work well. On CD19, it's a very specific marker. There are no antibodies to it, but what we're trying to do with the CD19 is to recognize those B cells that have CD19, so we can, in some ways, understand what the CD19 as an antigen is. Then we can draw the same person's T cells and make them target these B cells that are expressing CD19. So I would take T cells from you. If you have a B cell cancer, I would infect the T cell using a virus with an ability to form the receptor to the CD19, to target the CD19, or in some ways, it's almost like giving you a vaccination for the B cell, to recognize those B cells. And these T cells can now be proliferated, so I can grow them in vitro, so I can make them grow 1,000 times, 1,000,000 times, and I can put it back into your blood. So now I can increase the stoichiometry of the T cells so that they can match the B cells. So what do? These T cells will hopefully go and bind to these T cells and deplete them. So we've done this in many subjects in international clinical studies. We have over 50 patients, many of them pediatric, because this disease, acute myelogenous leukemia, and a few other subtypes of BALLs are very pediatric diseases. And these are boys and girls who are 8 years old, 10 years old, that have either they have to go through toxic chemotherapy with a very low life expectancy, or they have an opportunity to enlist in a trial like this. And we have some of them that have been almost in remission for 2 years. And we are trying to figure out what are the footprints or fingerprints of this remission, how we can find out who those patients are that can be eligible for this kind of therapy. So this therapy is evolving, and we are getting better at it. I think our first set of T cells that we were targeting against the CD19 cells had a low life, that they only lasted a few months, so we had to re-inject you with these. So it was not like I give you one injection of T cells or one infusion and you have a cure. So we have to repeat the therapy many times. Now we are coming up with further signaling domains that can be attached to the chimeric antigen receptor, and these signaling domains sort of enhance the ability of these T cells to even find the last B cell, to not leave any cell left. Because even if you leave a couple of B cells, they can replicate and they can form the B cell lymphoma again. So you need to get to a kind of total control on this. And we're trying to make this technology better with multiple generations. The promise is fantastic. The approval for these the ethical approval for taking cells from humans, manufacturing them in a laboratory, and then giving it back to the clinic to infuse these patients with your own T cells that have been infected with certain type of virus that can manufacture the chimeric antigen receptor. The approval of all of these is complex. So this therapy is not going to be cheap, because I've asked our own labs how many people are working to, for example, make one person's T cell library attack their B cells. And there are over 20 people working for a few weeks. So it takes a few weeks of manufacturing and proliferation of these cells and releasing them to say that this is now ready for treatment. It's complex, but the challenge is fantastic. You are now providing new life to these patients, who otherwise would see a sort of a terminal situation where they don't have a life expectancy that long. And the only things that are stopping us here, as I said before, is the cost and who's going to pay for it, and our own creativity, how much we can get on this. Now, there are further ways to perfect this. I mean, before I go in there, this technology is so attractive. There are many biotechs. I don't know if there are any in Russia, but there are many biotechs, and this is just a snapshot from 2015. And over the last 2 years, there have been more biotechs that have formed deals with the pharma companies using this technology, because you could have a few people who understand how to do cell cytometry, extract cells with CD19, infect T cells with the particular virus antigen that can manufacture this. It's not complex, so people who are skilled at labs can do it. So many companies have tried to attempt solutions for doing this, and they want to partner with pharmaceutical companies to conduct clinical trials and bring them to the market in some ways. Now, the final part of my talk is now, what are the other new technologies that are available for us for cure? So if I go back to this technology of using T cells to attack the B cells, we're still using some things like viruses to infect the T cell, because that is the only way we can carry a piece of genome into the T cell and to make the T cells express it. And that's a well-known technology, but still the viruses can become active, because we use, in some ways, suppressed adenoviruses or HSV, simplex viruses, or other virus technologies to infect the cell. Now, the viruses have been around for a long time, yes? And there is a way that bacteria has developed a technology as a primitive immune system to prevent them from virus attacks. And how do they do it? And this is something that you may have heard about, called CRISPR, or clustered regularly interspaced short palindromic repeats. I think nobody knows what this is, but they all know CRISPR. And I will try to tell in a few minutes what I understand from this. So, you can think of this as a bacterium being infected by a virus. And these viruses are called phages, and they live in bacteria. They inject their genome into the bacteria, use the bacteria's biochemical laboratory to make more of the virus genome, and then break the bacteria and come out, and there are more viruses. And now, what does the bacteria try to do? Evolution is fantastic. They try to recognize these viruses, to take some part of their DNA and put it into their own DNA. And they have used a mechanism of cutting some of the virus DNA and putting it into some sequence, which we call as CRISPR today. So in future, as these bacteria divide and evolve, in future, if they have another infection from a similar virus, they can actually recognize the virus genome, and they are ready with some kind of CRISPR RNA that can cleave that genome. So essentially break down the virus genome and prevent it from replicating. So it is almost like the first virus infection of the bacteria injected the bacteria with a vaccination. And now the bacteria is immune to future attacks from the virus. But this is a primitive immune system. Now, what can we learn from it? We are trying to use this technology of CRISPR to use human cells to make targeted deletions or targeted insertions into the human genome. And we're using this technology using this molecule called caspase-9, which is a ribosome with a guide RNA to make. So if you imagine this as genomic DNA, and we know, say, for example, there are many babies today born with certain diseases that are a single gene disease, like cystic fibrosis. We know it's a single gene that encodes a chloride channel, I believe, and that results in pulmonary dysfunction, and these people don't last very long. And we are using different forms of gene therapy where you can, inhale plasmids that are made with viruses that can encode the normal type of this chloride channel, and you can, in the pulmonary epithelium, you can transfect these genes where they are expressing the normal chloride channel. But you can do it at a genomic level. You can take the embryo, if you know this baby is going to have cystic fibrosis, and with a single insertion, you can insert the right gene to make a normal chloride channel instead of a defective channel. So thereby, you can have the whole genome fully changed. Or you can do it in those parts at a stage of a blastocyst, or where you know that this bud is going to form the pulmonary epithelium. Now, this technology is extremely powerful, and what draws us, what keeps us from simply practicing this is the ethics itself. Now, there are some ways that I understand today that you can actually use this technology to make your own organs if you need. So, for example, I can take a normally growing pig, and pigs are in some ways somewhat similar to the physiology of us for size, etc. We use pig heart valves to do surgery on human hearts and replace valves. So you can develop a pig that will eventually develop a human pancreas by changing a couple of genes in the embryonic stage of the pig. So when the pig develops into a whole animal, it can have a human pancreas. Now, those of us with diabetes or type 1 diabetes who have dysfunctional pancreas can have a complete resection, and you can have your own custom-made pancreas from a developed pig and implant it into yourself. Now, ethics stop us from doing this because we don't know if this pig could actually develop a human brain, because some of the technology that we are doing to make these targeted insertions or deletions have the ability to have transient mutations, or to change other organs where you don't want the desired effect. So there are many things that stop us from doing it, but the technology itself is beautiful, and it can lead to a level of personalization or cure, if you will, for these diseases that will make them almost be absent. You can develop humans in future that have no signs of these diseases that are eventually tracked back to a single part of the gene. So that's the promise. I mean, I guess all of us working in the science area are all trying to, in some ways, address different diseases, but the eventual goal is to prolong life. And some of these technologies like CRISPR, the abilities to use pluripotent stem cells to develop new organs, to regenerate tissue. Those with myocardial infarction can have new heart tissue. You can insert stem cells into the heart today and develop, better heart tissue or regenerate the myocardium. There are people who are elderly who are losing muscle, and you can put some of these stem cells into their muscles, and they can develop new muscle. So many of these technologies will eventually help us live long. And one of the best examples that I found of why people are so interested in this, it's not just biochemical scientists. It is, in fact, just true scientists. So there was a new company formed called Calico. A number of people, originally formed by Google, but a number of people working in pharma industry, bioinformatics, computational scientists, biochemical scientists, somebody from Novartis, from Roche, Genentech. Many of them have gone to join Calico. And the goal of Calico is to eventually stop aging. I don't know if we will stop it, but we could delay it at least. And maybe we could live 150 years. So the promise is huge. I think what stops us is our own creativity and how we fund these technologies. But the tools that we have today at your disposal, and when you grow up into being in industry and in big positions, you will have a lot more technology at your disposal. And what will just stop you is who's going to pay for it and what's your creativity in this. So thank you for your attention, and happy to take any questions.
Thank you very much for a very interesting lecture. So I hope there are some questions. Ребят, если хотите, можно по-русски, можно по-английски, как удобно. Давайте задавать вопросы.
Thank you for your lecture. So here is my question. Do you have, Novartis, do you have a production in Russia? Production factories?
Yes. We have one in St. Petersburg. It's called Neva. It's in a special zone near the Neva River, north of St. Petersburg. And we have the first licensed batches this year. And hopefully we will be able to provide medicines from Neva plant into patients in Russia. We are manufacturing in Neva. About 300, I imagine, at this point. Welcome.
Thanks a lot for your lecture. So the question is, what's your attitude to bacteriophages as for an alternative to antibiotics?
Bacteriophage.
Yes. I don't know whether it's on the world market, but in Russia we can see a lot of drugs containing bacteria that claim to work as antibiotic, as an alternative to antibiotics.
Yes.
What's your attitude for that?
I don't have a specific opinion on that. I think the whole world of antibiotics is challenging because evolutionarily you don't want to hit bacteria with huge doses of antibiotics because bacteria are very rapidly evolving and they can find resistance. They can generate resistance to specific antibiotics very quickly. Bacteria divides every 20 minutes under the right conditions, much faster than any of our cells can divide. So we need to have a balanced approach every time we take antibiotics. And it's a judgment call on the physician on the lives they need to save and what is the escalation in antibiotics. You always start with some antibiotics that is wide and then try to go to a narrow spectrum antibiotic. Now the bacteriophages, they are not working in a way like the antibiotics. They are working in a more gradual way. So if you want to save somebody from acute septicemia, I don't think you will give them a bacteriophage because they will likely not survive with that. So you want to give them a powerful antibiotic that stops the bacteria from growing. But if you have a common cold, for example, you get frequent cold and you think it's bacterial infection and you take frequent antibiotics, you will develop resistance, quite surely. So at that time, for somewhat benign diseases, you can think about more comprehensive therapy and how to leverage some things like bacteriophage therapy. But acute sepsis, bacterial infection, growing every 20 minutes, going to take your life, you have to use antibiotics.
Thanks a lot. And may I ask one more question? So I know that Novartis popped up after Sandoz and Ciba merged.
Yes.
So and what scientific policy did you get from Sandoz or from Ciba? And what has changed since that time?
So I don't know if it is scientific policy, but Sandoz and Ciba were both chemical companies. Sandoz made big chemistries. They had a division in fertilizer. They made fertilizer for plants. They made dyes for fabrics. They made medicines. Ciba also was a big chemical company. Also many of these. When Sandoz and Ciba merged, we spun out the fertilizer company into Syngenta. Then the other chemical companies were spun out differently. And what remained, Novartis, became more focused not on just chemistry, but on developing medicines, on biology, understanding physiology. So we have a street inside Novartis in Switzerland, which is the headquarters. It is called Fabrikstrasse. And Fabrikstrasse is where we used to make dyes for fabrics. But today, there are no dyes being made there. They are all biological scientists who are working on immune diseases. They're working on high-throughput screening. They're working on developing new antibodies, transgenic mice, all of these. So historically, what have remained is the chemistry has gone off into more of the fertilizer-type companies, and what's remained with us is more biological, genomic, physiological research. There is chemistry too. It's only at the part of finding a new medicine, but understanding pathways is less of chemistry, more of biochemistry.
Yeah, Ham here. So thank you for your talk. And I was wondering about the CAR T-cell technology. The point is that there are different types of T-cells. So I was wondering which type of T-cells do improve. For example, is this T killers or Th0 or Th1 to use in the therapy?
I don't know the details of that. I think, we have to, there is no rule of this is the type of T-cell we can use. We need to understand what is the best for those types of B cells, depending on the receptor, if it's CD19 or CD20 or CD52, what it is, we need to understand which T cells are able to do the job. And most of them, I would assume, would be NK-type T cells that have the ability to kill. Now, there are other technologies where you don't need to use these T cells. You can go with a conjugation approach, where you have an antibody loaded with a toxin that, or sometimes radiotoxin, that can just kill the cell without a T cell. There are those we can use as well. So there are many choices, and it depends on the type of B cell. and the type of disease and the speed of proliferation of those cells.
Got you. Thank you.
Thanks.
All right. So,
Yes, Alexander.
thank you for your talk, indeed. And my question is a bit different from the scientific questions that we had. So, my personal experience of interactions with big pharma, although limited as it is, in terms of personalized medicine, is such that the very moment you pronounce the word personalized, they go, no, 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 no. So, but it seems that it's a bit different. So, as of an insider, what is your attitude, let's say, and what is your feeling towards personalized medicine?
Yeah. So I can understand that response of why we say no, no, no. Because if you look at medicines and how pharmaceuticals work, it's highly regulated. So we have a rule book of how to apply for a new medicine with the Ministry of Health and how it's approved. And this says exactly how we need to do clinical trials, what we need to demonstrate. And this rule book today has very little about personalization. So the way we can approve new medicines is through randomized clinical trials, and they need to be done for diseases that are available in the ICD-10 library. And the ICD-10 library has nothing about immunoglobulin E associated asthma. It's asthma. So we need to approve medicines for asthma. And we need to approve it for the whole population. Otherwise, we will be considered as serving narrow interests on this. So what we are trying to do as a company who is interested in the science, who understands this, is to work with the regulators to change the way we can approve new medicines. And I can give you an example where we are trying to do this in asthma. It's not with this one that I showed, the NIGI equipment. It is for a new molecule which actually targets interleukin-13 receptor. And we've found this in a clinical trial that exactly the way I showed it, there are some people who do not respond to this medicine, while there are some whose asthma is completely controlled. And it depends on a pharmacogenetic mutation in the receptor type of interleukin-13. Now we have taken this medicine and we've gone with the European Medicines Agency and FDA to say, can we approve this medicine for those people who have this pharmacogenetic mutation in asthma? Because for them, it's 100% efficacy. For the others, it's not. And we are still in negotiation with them. And so we don't have a guarantee that we will do it because we need to do the study and we need to make it practical for doctors that are treating asthma. We cannot explain the whole interleukin-13 receptor morphology and pharmacogenomic variations to these doctors. We need to come up with a practical test that they can do and identify who those patients are. So once we have the full solution, I think we will be able to do more in personalized medicine. But the challenge, as you see, is in the regulation and the practice of medicine. It's not just changing the biology or changing the medicine, changing the practice takes longer time.
Thank you. I think we'll finish with the questions, and I think Krish, let's thank once more Krish for a perfect lecture. Okay, we'll now have a break for 15 minutes, and also if you have some more questions, I think Krish will be here like for 10-15 minutes more.
Yeah, so regulators and